Good evening, everyone. I would like to bring this monthly meeting of the Entomological Society of Washington to order. Recording Secretary Gary Neville, could you please read the minutes of the last regular meeting?
Yes. The 1234th regular meeting of the Entomological Society of Washington was divided between an in-person group at room 20, WG 33 in the National Museum of Natural History and a virtual audience. As President Matt Buffington opened the meeting at 7 p.m. on the 2nd of February, it was noted that the total visitors were 63, with 18 at the museum and 45 virtually. The first agenda item was a reading of the minutes of the last meeting by Recording Secretary Gary Neville, and these were approved. Next, Membership and Communications Secretary Elizabeth Young announced the official membership of Michael Bernal and Laura Williamfor, with Eliza Veta Pliz as a new applicant for membership. President Buffington noted that the new ESW website is close to a debut, with further information on this soon. He also announced that the ESW Executive Committee, during a recent meeting, has approved an amendment to the bylaws to establish a new website administrator to serve on the Executive Committee. Members of the Society will vote on this new position during the March meeting. President-elect Don Weber announced that the Entomological Society of America will sponsor a free tour of the entomological collections in the American Museum of Natural History at 10 o'clock a.m. on March the 4th, conducted by Jessica Ware. Program Alan Norbaum noted that the speaker for the March meeting will be Andrew Jansen. Anand Davis suggested an improvement for the January 2023 minutes to add details of the date of mid-November 2023 for the ESA meetings in Oxon Hill, Maryland. Program Chairman Norbaum introduced the speaker of the evening, Dr. Robert Kimsley of the University of California, who presented forensic entomology and its potential role in fire death investigation. Dr. Kimsley's research interests include public health entomology, arthropods of medical importance, zoonotic disease biology and ecology of tick-borne pathogens and tick-feeding behavior and biochemistry. He has gained a reputation as a forensic entomologist and has assisted in more than 300 criminal investigations. He outlined a particular case in which he successfully provided flesh fly lifespan information for it. Dr. Kimsley presented step-by-step procedures for a few criminal cases in which forensic evidence was strongly important. He has taught a week-long forensic entomology course during several years with an average of 70 students. The first part of each course involves lectures followed by field work. In the field, various scenarios are presented, such as car crashes, fires, gunshot deaths, and many other situations, using real human corpses. The human bodies are supplied by a company in Tennessee that respectfully gathers them from relatives. After use in the field work, the bodies are properly cremated, then returned to the relatives. Students analyze timing of fly appearances to bodies and the growth sequences, then present their findings in classrooms. After the presentation, there were many questions from audience members. The meeting was adjourned at 8:40 p.m.
Thank you. Recording Secretary Gary Neville, does anyone have any corrections to the minutes as they were read? I get a motion to accept the minutes as they were read. Is there a second for that? Second. Motion carried. Thank you, Recording Secretary Gary Neville. We have a number of absentees tonight. That would be past president, president-elect, secretary.
What do you mean absentees?
Oh, sorry, not present.
Oh, okay.
Person, sorry. Thank you for pointing that out. So we're gonna, yes, sir. Well, the issue we're having here is, for everyone here in WG 33, the camera is not pointed at me, it's pointed at y'all. So, keep that in mind. So the, one thing I would like to point out, indeed, we were going to have a vote on the bylaws. We're updating the bylaws to include a website administrator. As it turns out, there were a number of other things in the bylaws that also need addressing. So instead of sending out piecemeal articles for the membership to either yay or nay on, I decided we're just going to do the whole thing at once. And there's some articles that we don't adhere to anymore. There's a few typos, and we're just going to do it all at once. Okay. So hold that thought. We'll be getting on that maybe next meeting or the meeting after. Are there any visitors that would like to introduce themselves tonight? Or if anyone would like to introduce said visitor because they're a little shy and they don't want to introduce themselves.
I can introduce Spencer Oates. He's an intern.
Okay.
He's a Cornell graduate. He reads University of Hawaii.
Awesome. Welcome.
Well,
I guess I was here last time, but I didn't introduce myself. I'm Estefania. I'm a pre-doc fellow working with these children.
Anyone online out there in TV land that would like to say something? Alyssa says, welcome, everyone. Okay, I don't hear anything. Boy, we got a major Don Weber presence in this room. This is great.
What's that supposed to mean?
Well, you're like on the screen. I'm not sure how this is happening.
Oh, okay.
I think it's by the speaker. He last spoke.
Yeah.
he's not.
Well, those cicada wings are pretty big. Do you like those? I just put it in my office.
It's a
Does anyone have any exhibits that they would like to present this evening or announcements for the benefit of the society? Anyone out there in TV land like to say something? Or we're just moving this meeting right along. Everyone's excited to hear Andrew. Without any further ado.
Hey, Matt, I thought you were going to call on me for my
Oh, that's right.
but I guess that's a type of announcement.
Sorry about that.
I have a partial update on the June ESW Banquet. I do not yet have a venue. I have a world-class speaker, but confirmed. But I want to take an informal poll right now, or you can also follow up with me by email or in the chat. You know, in the past, we've done potlucks. Some venues lend themselves to that, some don't. And some people are more in favor of sit me down and bring me my food. So the latter costs some money, but you don't have to cook. And the former is cheaper, possibly more interesting, and it requires you to bring something. So what I want to know, because they've been getting mixed messages, literally, from different people, what is the member preference, assuming you have some tendency to come to the banquet in June. We're shooting for Thursday, June 15th. What are your preferences in terms of that format? And I don't know if we want to do an informal poll right now, or you want to sleep on that, or what? What do you think?
The banquet's planned to be held in the evening. We're not doing the lunchtime one again.
That's correct.
Oh, yeah.
Everybody's around.
At least 15.
Do we have any kind of advocates on one side or the other who want to voice their opinions? Or do you want to pepper me with emails or chats? Or do you want to, does anyone think it's appropriate at this point to take a straw poll of sorts?
You'd be taking a straw poll of 12 people.
Yeah, that's not a lot, I know.
I
But
vote
your
for-
voice matters here.
Okay, I vote for the most expensive option. I won't be there.
Who's that who's in favor of the most expensive option? How are
Wait, Lourdes just said there was a problem with the link, so a lot of people aren't in yet. Should we wait a second?
Uh-oh.
Sure.
Oh, there's only 17.
Let me see if is that the case? It got fixed?
Hi, yeah, the link on the website, I tried it originally and it was telling me that there was already a meeting going on, so I just fixed it.
Oh, thank
but
you.
I'm gonna try and get on again through my phone. But that's probably why the count is a bit low than usual. I'll check on that.
It's not the subject matter. Andrew.
Yeah, nothing on you, Andrew.
Well, I'd say for the catered, not catered, potluck choice, I'd say probably the best thing would be to email Dawn if you have a specific preference. Is that okay, Dawn?
Yeah, and if you don't email me, I'll assume you don't have a strong feeling one way or the other.
I would agree.
And right now, I'm looking at a couple venues that are walking to Ballston Metro in Arlington, but I haven't firmed them up, so we're still looking at things.
There was an Italian place we used to go to there.
I don't know about that. You can tell me about that. You can email me that.
Okay. I don't think it's bulldozed yet. It was
Okay.
It was old school. Okay, it was nice.
All right.
All right, thanks, Don. I appreciate the.
Thanks, everybody. Send me your druthers. The only thing I can promise is not everybody's going to be happy, but you're going to be happy with the speaker, so that's the most important thing.
Cool. All right. Thank you. Okay, I think now, are you ready to introduce the presenter?
Excuse me, Matt.
Who was that?
This is Lourdes again. Sorry. It looks like the camera is facing the audience. Is that?
Yeah, and I cannot figure out how to assign the other camera, which is the one that's normally on. It doesn't show up in the option list. It's only the camera on the screen facing the audience.
I see. Okay.
Should we have
Remember how we fixed that last time? I feel like we had that problem last
You
time.
probably unplug it and plug it would be the first try.
You can choose from different cameras, but I don't see there's only one choice and it's the one that's activated.
If there's a button on the camera.
Maybe, is there a way
Maybe
to,
it's turned off.
is it a way to turn it on?
Or we just move the webcam from the big screen onto the little one.
Or we can make
Put it in his face.
Andrew sit over there and
We all sit over there.
and I'll push the button up here.
But they won't see the screen.
No, they won't see the screen. Well, can we share the screen from this machine? Isn't that normally?
Yeah,
how
we
do
usually do that, yeah.
Yeah. Okay, so they'll see the screen.
They
They
just
won't see Andrew,
yeah.
but they'll hear you.
It's okay.
Okay. All right. Ready to go. All right.
Okay, our speaker tonight is Dr. Andrew Jansen. He received his PhD in evolutionary biology from Arizona State University, working with Nico Franz. Although his academic background was strictly in entomology, he has a BS from the University of Florida and a master's from Arizona State. His interest in cuticle microstructure led him to study the evolution and biomechanics of the exaggerated rostrum in the genus curculio. That was the topic for his PhD at Arizona State. As a postdoctoral fellow at the University of Bonn, he studied the influence of mesoscale cuticle laminate structure on the evolution of head shape in Polyneoptera. His goal as a researcher is to perform systematic study of cuticle layering patterns and to evaluate cuticle microstructure as a phylogenetically informative characteristic. Andrew. Thanks for the introduction. Who wrote it?
Just like that.
What are you talking?
Boy, oh boy. Is that okay? Wait, just share it on the Zoom. Yeah, that'd be this one. Participants can now see your application. Cool. Can everybody see the presenter, the presentation and presentation?
Ellen can see it there.
Okay, great. So this is kind of a talk that I guess you could say sums up some of my thoughts that I've been having on the subject of, I guess, dealing with insect cuticle. Most studies that have come out on the topic of cuticle have been engineers that are interested in the mechanical principles of the cuticle and the way that insects are able to manipulate their exoskeleton to do some pretty incredible things. However, we still don't know yet enough about cuticle and about the homology of the different parts of the cuticle, and there are different parts, which I'll show here in a moment, in order to enable like broad scale comparative studies that people are starting to attempt. And this has led to some very interesting problems, which I'll just kind of show you here in a moment. Now, looking at the slightly antagonistic title of this, you might think to yourself, well, looking at insect cuticle, what about it exactly would engineers or biomechanists actually care about? And cuticle is partly they're interested in microstructure and partly in mechanical performance. Cuticle is what's called a hierarchically structured material. It is built of essentially two components. So there is a fiber and then there is a matrix. The fiber is chitin. Chitin is a kind of sugar and it's super duper brittle, but very, very stiff. So to take advantage of the stiffness while mitigating the brittleness, they wrap these chitin fibers in protein and then embed that into an amorphous protein matrix. And in doing so, they form sheets of chitin that are a little bit less brittle than pure chitin by itself. The other trick that they have up their sleeve is they then arrange these chitin fibers into these layers of chitin fibers into a twisted helix. So this is called a helicoidally arranged ply. So you have sheets of chitin that are deposited or grown from the epidermis at a slight angle compared to the previous layer. This seems to be tied to circadian rhythm. That seems to be the way that they're actually achieving this deposition. And in doing so, they get this very interesting looking laminate that is simultaneously quite stiff and also very tough. It's quite hard to break these. Anybody who's ever tried to step on a cockroach knows that when you step on them, their guts come out, but the exoskeleton doesn't break, right? And that's because that's basically the toughest part of their body. And one thing I want to emphasize here is that the exoskeleton is not completely uniform all the way through its thickness. If you look at it in cross section, so imagine if I took a piece of let's say a lobster, this is actually a lobster cuticle, and I broke it in half and then looked at the broken end of it, what you can see is that there are some distinct zones or regions within the cuticle itself. So towards the outside of the organism, here on the right side of the image, we have the outermost layer or outermost region or zone called the exocuticle, and then the innermost zone is called the endocutical. These regions can be approximately uniform through their thickness, but relative to each other, they're quite distinct. And in fact, you can see that in this particular individual, there is a very sharp dividing line between the two. Their mechanical properties are different, their composition is different, And some features such as porosity or sclerotization can also be very different between these two regions. In some cases, there is a third region called mesocuticle that's not very well defined. We'll get into that here in a minute. But just know for the moment that there are distinct zones within the cuticle, and that seems to be a subject of interest for people who study these animals. So I'll show you a couple of examples of things that sort of typically get studied when somebody examines insect cuticle. This is a very famous example, a quite recent one actually. This was this is a study of ironclad beetles. Anybody who's ever tried to pin one of these knows exactly what happens. You end up doing that to your pins, and the beetle laughs at you. These things, there are anecdotes about people running them over with cars and finding them like, just, they get up and crawl away, no problem. And in fact, when they tried crushing one, and I'm not joking, this got published in Nature, they took a beetle, put it between two plates and squeezed it until the exoskeleton cracked. That was their experiment. And what they found is that, yeah, it actually could withstand the force of a human being standing on it. That's how strong these exoskeletons are. And when they examined this a bit more closely, they found that there were some morphological features of the elytra that enabled them to withstand, or to distribute force more evenly along the wing covers. And then also the cuticle, which you can see obviously has layers, is layered in a particular way that makes this a bit more robust. Another cool example that has gotten a lot of attention by folks in biomechanics and engineering is this critter right here, the peacock mantis shrimp, and in particular its dactyl clubs have received a lot of attention. So I'll show you a video. If you don't know what this is, I'll show you a very short little video here of the behavior that they're interested in. Here's our little guy, and you see these two clubs right here. It will deploy them to actually crack or fracture a target. So you can see what happens right there. It's extremely fast. You blink and you miss it. It has the accelerating velocity of a bullet. So they say, oh, it hits it with the force of a bullet. It has the accelerating velocity of a gunshot. So it's very fast. It actually causes cavitation to happen. So bubble forms and then implodes, and that shock wave is what actually fractures the target. But of course, the question is, how do they fracture the target with this and not break themselves? I mean, after all, they are made of chitin. So the way that they do this appears to be a pretty sophisticated mechanism for preventing cracks from forming. So this is a paper that was published in 2020. I think this one was out of Purdue, as is the next one. And you can see that a lot of thought has gone into the micromechanics of this cuticle structure. So very interesting topic. They found that cracks are unable to form in this material, or it's much more difficult for them to form in here. So it seems that this twisting architecture sort of prevents cracks from happening and mitigates brittleness in the cuticle. That's what gets most of the attention, except when this isn't the case. So one example that I studied was this group of beetles, the genus curculio, this curculiosae, a female, super long rostrum, really awesome behavior. They drill into acorns and they bend the snout. The snout is actually a cylinder. It's hollow on the inside. All of the tendons for the mouth parts, the esophagus, everything else, nerves, runs all the way down to the apex where the mouth parts are physically located. The muscles that drive the mouth parts are actually located in the head capsule all the way towards the back. And what's interesting is this behavior right here. I ran this experiment at ASU, and I had this just on repeat. The same, so this is a piece of ripstop nylon. It has very little, it doesn't fatigue. It's very, has high tensile strength. I ran this experiment and they told me, just come back in a few days when it's broken. 2 weeks later, they asked me if I could stop the experiment because it was running up the power bill in their test machine. So this thing ran 400,000 times before I stopped the experiment. 400,000 cycles of bending with no damage to the structure whatsoever, which is pretty impressive. If you took this beetle's leg and tried to do the same thing, it would snap on the first attempt, right? I mean, you can see how extreme the curvature is here. You know, it's 90 degrees and then being flexed until it's completely straight. So there's something going on here that enables that to happen. And without getting into the nitty-gritty of it, the basic mechanism is that the cuticle in the apical portion of the snout is optimized for bending. It has a very thin exocuticle, very thick, beefy endocuticle. Endocuticle is usually softer and more compliant than the exocuticle. Exocuticle is very stiff. And then the basal part is optimized, or appears to be optimized, for rigidity, so it doesn't bend. And this results in a very even distribution of stress when the bending behavior happens. There's some other things like fiber orientation is different. Beetle endocuticles have lots of fibers that are very large and easily seen, and they're optimized for bending in the apical part and not so much in the basal part. So you're probably thinking to yourself, okay, well, you've convinced me that these are all engineering problems, right? Here's an example of something. This is Heteropteryx dilatata, big banana-shaped phasmids, right? Interesting critters. A guy in 1992, his name was Eric Loader, did a study on their cuticle development. He's interested in the genetics of cuticle development. He broke open all of the parts of this insect's body and looked at the composite structure in all of those organs. And he found that every single one of them had a different layout, different layer thicknesses, different numbers of layers, different fiber orientation angles, and different total thickness of the laminate. All of those components do something to the stiffness and the toughness of the resulting structure, but nobody knows what. And the guy wasn't interested in that. He just published this image and described in a little more detail the laminate structure in each body part. If I were an engineer and wanted to examine this system in a little bit more detail, studying this within a single organism would be pretty easy. If I wanted to compare, say, the leg to the pronotum, right? Where would I start? Well, I could do what this guy did. Bart in 1972, I believe. He basically looked at different body parts and characterized the thickness and number of layers of endocuticle, exocutical, and mesocuticle. So within a single organism, it's relatively easy to identify these zones, right? The endocuticle looks pretty much the same in a leg as it does in, say, the pronotum. It's when you try to make comparisons between species that it gets a little bit more dicey. So, for instance, if I have these three Polyneopteran taxa, a mole cricket, a rather large stick bug, and then a cursorial praying mantis, one that actually chases its prey, which is pretty neat. And I wanted to compare the cuticle in the head. How do I know which regions of cuticle are the same? In other words, how do I assess homology in the different layers of the cuticle? How do I know that mesocuticle in one taxon is derived structurally in the same way as mesocuticle or endocuticle in another taxon? How do I know what things are evolutionary novelties or not? And the answer is there isn't an answer. Nobody knows. It hasn't been done yet. The last time that any sort of layering homology was done in cuticle was in 1952 by a guy named Richardson, and I believe it was either his tech or his student, Leo Schatz. And they came up with a scheme that was based entirely on staining. So you would take a piece of cuticle, cut it. It had to be preserved in Bowen's fluid first, so you're already messing with the protein structure. You section it, put it on a glass slide, stain it with Mallory's trichrome, and then whatever color it is, that's what they're calling the layer. And importantly, this was completely divorced from any notion of structural homology. So it didn't matter what the structure looked, what kind of structure it had, if it was helicoidal, unidirectional, where it was in the land, that didn't matter. If it was colored that way, that's what it was. If it stained yellow, it was exocuticle. Didn't matter if it was all the way on the inside of the organism, right? That was the way that they They were very consistent and very adamant about that. Nowadays, informally, when people do, say, SEM or TEM, there is no staining involved, right? So generally what's accepted is that if you see helicoidally arranged flies, that's exocutical. If the fibers are all facing in the same direction, we call that endocutical. That's the scheme. That's it. That's what we've got. It's terrible. And I'll show you why here in just a moment. So looking at these three species, if I cut open their exoskeletons and look at it, let's just say with TEM, no staining, no coloring. Where's my mesocuticle? Where does my exocuticle end and my endocuticle begin? I don't know. It's pretty hard to tell from just this image, right? The layers are confluent. You could maybe say, well, this one here, it has a pretty clear division right about here, right? This part looks different from down here, but then what's all this down here? That looks a little different. What about these layers? Does it have multiple mesocuticles? What do we call this? And there's no answer for that at present. If you try staining, the staining characteristics aren't very helpful. So in my eyes, I've annotated this the way I think it should be. You would have exocuticle be this mostly helicoidal zone out toward the outside, and the rest of this is endocutical. I don't know what mesocuticle is supposed to be. It's very badly defined. But the staining characteristics of the outermost helicoidal zones are not consistent between species. In one species, it's yellow. In another species, it's pink. In the next one, it's blue or purple. When you stain with the same exact stain, we don't know what's being stained either. So this doesn't really work. And in fact, in the literature, and this is what's most concerning, engineers don't know the answer to this question either, and they don't have the biological background or training in histology to be able to make these distinctions. So you end up with things like this. Here's a piece of cuticle excised from Pachynoda marginata. This is a scarab beetle. And here we have a helicoidal zone, outermost region, and part of it stained a little bit pink and blue, so they called it endocutical, even though it's on the outside of the organism, which makes no sense. And it's also confluent with these other regions. It makes no sense whatsoever. And the same thing happens down here. There were layers that were, yellow, and they said, yeah, that's exocutical again, even though the structure is obviously very different. Along comes a biologist, Thomas Van de Kamp, who looked at beetles and was able to distinguish rather quickly, the same way that you or I might as biologists, that no, there is an outermost zone, which we can call exocutical, and an innermost zone comprised of very distinct layers, very fibrous layers, that then would be considered endocutical, excuse me. And he came up with this whole scheme for how this could be developed. He created some concept of homology just for beetles. This doesn't necessarily apply to things outside of Coleoptera. But it took a biologist to do that. And these kinds of studies, this is the only study I know of where this has been done. And this is what enabled me to do a PhD dissertation on my taxon of interest. And here it is in curculio. You can see it very easily. There's an outermost zone here. This is a fractured rostrum. So you're looking at the cuticle here in cross section. Here's our exocuticle, which fractures one particular way. It looks very smooth. And then this rough fibrous inner region, here's your endocuticle. A large number of layers of very large, well-defined, they're called macrofibers. So, okay, the staining thing maybe isn't working so well. What if we try the sort of informal structural definition that exists? Maybe that will help. Here's another study published by Schmidt et al. This is out of the lab of Stanislaus Gorb, who does a lot of really wonderful mechanical studies on insect body parts. He wrote a book on it, in fact. But here, when they tried to define what the different parts of the cuticle were, they ran into a little bit of a problem in my eyes. Here we have a helicoidal zone on the outside that they're calling exocuticle, fine. a unidirectional region here. All the fibers are facing in the same direction, and that's why it looks like that when it breaks. It's fairly uniform. I call that mesocuticle. So far, so good. And then we run into more helicoidal plies. So they called it exocutical, because currently that's how it's defined. If it's helicoidal, it's exocutical. And then the remaining unidirectional plies, they said, are endocutical. So there's this really strange arrangement. And this, how do I know if I want to compare this organism to, say, another species of phasmid that doesn't have this unidirectional stuff sort of sandwiched in here? How do I know that this corresponds, this presumptive exocuticle here corresponds to the outermost layer in another species? How do I know they're homologous, derived from shared ancestry? We don't. At the moment, there is no way of assessing that, or no way of that it's defined formally in the literature. There have been a couple of attempts to rectify this when these sort of unknown layers pop up. The best one that I saw was a guy named Anthony Charles Neville. This is in the mid-60s. He looked at cross sections of locust tibiae, and he saw that in some regions, the exocuticle had unidirectional layers in it, the same way as the endocuticle does. No reason they can't have both types of architecture, he reasoned. It's tied to their circadian rhythm. Maybe these are deposited at night in this particular location to enhance its stiffness. It seems to be made of the same material. It stains identically, but when you look at it with polarized light, you can see that it has a different type of microarchitecture. This study has not been mentioned since 1965. Even though it was quite a good one and it's referenced a lot for the circadian rhythm stuff, his discussion of microarchitecture and, you know, presumably the identity of these different layers is just not talked about. It's considered too crazy. So if I try this on my own samples, let's go back to the three things I wanted to compare. This is Eurekantha calcarata, this stick insect. So if I look here, I can see something that appears to be rather uniform with striations. If you look very closely with TEM, you see this helicoidal ply architecture. And it's the outermost layer. So towards the top is toward the outside of the insect. So you're looking at a cross section again. This could be an exocuticle, sure. And then you have a pretty uniform set of thick unidirectional laminae underneath of that towards the inside, endocuticle, right? Works for this particular location in this particular specimen. Here's the same species, same specimen actually, slightly different part of the head, closer towards the mandibular insertions. And in the middle of our exocuticle, we have not one, but two layers that are uniform in appearance and with TEM turned out to be unidirectional. All the fibers in these two zones indicated by the arrows are facing in the same direction. And what's more interesting is that you can see where they sort of peter out. These layers here are contiguous. So you can see what's sort of happened here. These layers are actually nested within the exocuticle, and they're part of it, I think. My hypothesis is that whatever mechanism is responsible for the twisting as the layers are being deposited, that mechanism is being simply arrested, and all of the layers subsequently produced are unidirectional until it's then reactivated and you get another zone with helicoidal plies. It's turned off and then back on. It's a very simple mechanism that could allow for that to happen. My hypothesis, not substantiated. But in this case, the structural definition where the helicoidal things are exocuticle, the innermost unidirectional layers are endocuticle, fails simply because there are exceptions to that. And I think the best solution for this, the solution that I settled on, has been to use a large number of different techniques. And I'm talking about electron and light microscopy, and then I tried a couple of other things like Raman spectroscopy, just to see what regions were actually of similar composition in terms of their raw ingredients, right? How much chitin, how much protein. And what I was able to find is that this region out here, even though it has these interesting looking unidirectional plies, actually comprises the exocuticle. So this whole thing is exocuticle. This whole thing down here is endocuticle. They stain the same. You can see where they sort of end, and it's a rather sharp line, which I find very interesting. And if we colorize those TEM images, this is how I would delineate the different zones of the cuticle. This is, again, my own hypothesis here. So there's a mix of both types of architectures within both of those zones. And I think that looks okay for these species. But more to the point, we need more studies like the ones that Van de Kamp did, where a large number of species are examined, their cuticle sectioned, stained, examined in SEM or TEM, to try and make these homology assessments across taxa, because otherwise it's impossible to make any meaningful comparisons. If I want to know if the endocuticle of a particular organism is responsible for, its stiffness, and compare that to some other species, right? This species has stiffer endocuticle than this other one. How do I know that I'm dealing with something that is derived from the same developmental mechanism? At present, I don't. And I don't think engineers are going to make that distinction for us. So this is sort of my way of saying I think we need more entomologists on this. It's an open field. Nobody is studying this at the moment. I mean, nobody. Like, I might be the only person, and then maybe one guy in Germany who's interested in this as well. So that's my shtick. That's what I'd like to study more, and that's what I think if you're young and going into grad school, you should consider looking at cuticle. There's a lot to see. I got a lot of help from a lot of people working on this, especially as a postdoc and as a grad student. I'm gonna end the talk here and open the floor for questions, but before I do, I'm going to very shamelessly talk about the electron and confocal microscopy unit, which is now again open after a brief hiatus. If you have insect specimens, you work here and have insect specimens that you would like to have examined with SEM, TEM, confocal, epifluorescence, and so much more, our doors are open. I'm here once or twice a week. You can find me on the 7th floor. Just talk to me. Happy to help. We're located on the Beltsville Agricultural Research Center campus. I'm down the hill in building 12. So with that, I'll take any questions you might have. Thank you. Yeah.
What is the benefit of distinguishing between endo and exocutical in the first place? It seems like these are kind of
artificial
distinctions.
They actually have different composition. They have different ratios of chitin to protein, and different proteins are involved. So what can be seen with, say, Raman spectroscopy is that there is a greater degree of, say, sclerotin activity in the outermost region than there is in the innermost region, or cross-linking that happens. So yeah, you can see that they actually have different mechanical properties if you perform different mechanical tests like nanoindentation. So they're actually distinct entities.
Okay, yeah. I sort of got the impression from your slides.
They look super uniform, but they're actually very different mechanically and very different chemically.
I mean, a lot of these were rather thick, like sclerites. What do you see when you're looking at like conjunctiva, right? Because going to the protein sort of ratios, right? It's like a higher resin concentration, like, oh, okay, this is
So one thing I didn't show is that the endocuticle has a much higher concentration of resin in it. If you put it under a confocal, the exocuticle is dark, but then the endocuticle glows bright blue.
Right,
which is pretty neat. So it looks like as you move towards like a conjunctiva or something, the exocuticle kind of peters out, and then the endocutical kind of just morphs into, there's a kind of a gradual transition, and then it's very abrupt where it becomes all resonant or mostly resonant.
So I mean, could you use then that, the conjunctiva and that sort of slope gradient as a way to like kind of with CLSM like trace back and go, okay, based on your the way the laser is bouncing back from the CLSM. like, okay, this is endo, this is exo.
That's actually a really good idea. I hadn't considered that. I guess in theory you could, yeah. I don't think anybody's actually done that. I've tried it before. I've seen people do confocal with the like with whole legs, and you can see where the conjunctiva is, but I've never seen that like in cross section myself.
You can do it with CLSF. Yeah, I mean, this is what we did with vagidial glands to show basically because I mean, for exocrine glands, right? It's just an imagination of the epidermis, and you can also get some cuticle that binds the resin of vagidial glands. So then you can track basically what's gland and what's epidermis and cuticle from inside.
Just good to see that. Maybe we should talk.
Yeah,
That's a good idea. See, entomologists figured that out in 5 seconds. I think Alan had his first.
Just curious, are there any developmental cues? Obviously, no one's done Evo, Devo, and cuticle development, right? Or
There is in Drosophila, because they've done everything in Drosophila.
But is there anything that indicate polarity of any kind of, where these different layers are coming from, et cetera, et cetera?
There seems to be some kind of a switch, where it says, okay, stop producing this mixture and switch to the next mixture. Most of the emphasis, though, on cuticle developmental research has been on the rotation mechanism. How are these things being deposited? How is every subsequent layer grown out at a different angle? And it's pretty cool looking. They image the surface of one of these epidermal cells. It looks like a pegboard. It's got just lots of these little pegs sticking out of it, and the fibers are sort of deposited between them at different angles. So for when each layer is, so that's where most of the emphasis that I've seen. Yeah.
These engineers you mentioned, what are they interested in using cuticle research for?
Most of the time, so that they can get into nature. But no, the main interest is in producing composites. So biomimetic composites are very difficult to work with because insects have what's called an asymmetric laminate. There's no symmetry about the midline. Those are notoriously difficult to get to behave the way you want them to. But when you can, they're very powerful. They let you do things that normal materials like steel wouldn't let you do. For instance, there was an airplane built. It was Air Force X5. I want to say it was like X512 or something. It was an experimental aircraft, and they made an asymmetric laminate in the wings so that when it sped up, its wings would start tilting and make it more aerodynamic just from friction alone. So no mechanical parts involved. It would just do that. And as it sped up, its aerodynamics would match the speed that it was going for fuel efficiency. Pretty cool.
It's a question.
Question in the chat. Walter, muted. There you go.
Oh, yes, sure. Thanks for calling on me. So I'm always interested in applied science, and this was a very interesting lecture. And I was wondering whether your research findings or even your methods can be used to construct inexpensive homes in third world countries.
So I actually got asked that question by one of the guys on my committee in ASU. He builds stuff with composites. What is it, Purdue? And the thought that they had was, this is sort of like working with these composites. It's like working with fiberglass, right?
Right, yeah.
Brittle, but you can make it into something that's incredibly tough by embedding it in resin. There's some talk of whether this can be applied to 3D printing. So I don't know if you're thinking of these 3D printed houses or whatever that, they just come in and they deposit layer upon layer to actually like construct a concrete facade or something. There could be application, but frankly, we just don't know enough about how these things are controlled and mechanically what's actually going on. what are the rules that govern the use of these different layer types? Like why in one structure is there a unidirectional layer and then in another structure there's nothing? Like we have no idea. If we could figure that out, then yeah, it probably would have some pretty broad applicability to use of composites.
All right, thank you.
Alyssa. In the chat.
Hey, this was really, really cool. And I can't help but think, like you're saying, what we need is a huge comparative library to just look and find patterns. About how long does it take to achieve one of these images?
Oh boy. Well, as far as just embedding something, you have to procure the specimen, pop off Cuticle. The embedding process itself takes, I don't know, maybe a couple of days, but it takes as much effort to do one specimen as it does 100. You know, you do them in molds all at the same time. And then the sectioning has to happen. And sectioning is where it takes time. It's one specimen at a time, and it can take a while. Yeah, I would say to produce these images, it took me, for the 3 specimens, it took about a month of work.
Yeah, that's really cool. But definitely sounds like it should be in big batches. There should be more research in this. But my other question was, are we any good at replicating Cuticle artificially, or is that still way beyond us?
It's an interesting question. There have been some attempts to do that. There was a guy at Harvard, I can't remember his name. He was using digested shrimp cuticle, so he was getting chitosan, it's a form of chitin, out of shrimp exoskeletons, and then he was using that to make like plastic cups and stuff. But he did not make any of the layering structure. He was just taking the raw material and making plastic out of it. The same way you use cellulose or something like that to make a cup. He was doing the same thing with shrimp chitosan. Yeah, so we're not in a place where we can make the where we can replicate the layering yet.
Yeah, I can't imagine. It seems really, really complex. Thank you.
Oh, thanks. It'd be super useful for small robotic vehicles or something, but I don't speculative.
So I realize the jury's still out on just about all adult insects with this respect, but do you have any insights of what layering might be occurring in larvae or immature hemimetabolous insects that in a rule would be applicable across multiple groups?
So in the parts that are sclerotized, there does seem to be that helicoidal structure, but the rest of the body, all the soft parts, the layering structure isn't very well defined. Yeah, the more membranous stuff tends to be pretty amorphous. It actually resembles, if you look at a phylogenetic tree and go back towards like, you know, the tardigrades and onychophora, they have layered cuticles, but the individual layers, the chitin is just in all directions. It looks like velvet or something, and there's just fibers all over the place and all facing every which way. So it's very kind of floppy and soft, and it's sort of the same thing in larvae.
I'm not sure if this is like relevant, but has anyone looked at the structure of wing veins?
Yeah, there was. Who was doing that? I think that was also Stanislaus Gorb's lab that was looking at wing veination. But they weren't looking at microstructure though. They were just looking at the effects of stiffened veins on the flexing of the entire wing. A bit different. There's a couple of other groups that were doing that. I remember who off the top of my head though.
So how much is known about how cuticle is? Is it mostly from the inside, so the outer layers?
So yeah,
secreted first, or can stuff come out like through?
So it's all secreted from the inside out, basically. So the uppermost layer is deposited, and then layers are formed underneath. It's grown out. Some stuff does get secreted through the pores. The epicuticle gets secreted through pores. It's like a cement layer that forms on the outside of the insect I've never heard of this. It, yeah, secreted all through pores, but that's usually not of much mechanical significance, so it doesn't get a lot of attention.
I read someplace a while back that there are more than 100 different proteins in the cuticle mixed in with the chitin. You obviously know something about the chitins that you chose not to share with us tonight. How much can you tell us about what you know about the different localizations of different proteins?
So the protein structure is a big, huge question mark. That's not something that's ever been really fully characterized. The emphasis is almost always on chitin as the load-bearing component of the cuticle. But the thing with that is chitin is the load-bearing component in plane. Out of plane, it's actually the protein that's supporting most of loads that are applied to it, because chitin is not very stiff in the off-axis direction. But as for exactly what proteins are there, I think there's only been maybe 2 or 3 organisms where they've studied that, and one of them is Drosophila. So for most organisms, it's not known. We have sections, we can roughly guess the orientation of the chitin based on polarized light, things like that, or staining for TEM. But yeah, the protein structure is a huge mystery.
There's a number of histochemical stains other than Mallory's trichrome.
Yep.
That are quite specific for different types of molecules. I hesitate to suggest their use when you're using Raman spectroscopy. Why go to 19th century techniques when you're yeah.
I mean, for it, they do have some utility. I mean, Mallory's trichrome is the one that got the most use because it seemed to distinguish between something in the cuticle. Yeah, what I saw suggested was that the stain was actually adhering to, I guess, small gaps between the chitin strands that were cut during sectioning, and that's what they were adhering to. Some people say they're adhering to lipids or protein or whatever else that's in there, but the fact of the matter is it's still not really known. As far as more specific histochemical staining, I did see somebody do immunostaining for resilin, which I thought was really wild. But I'm not really sure if you can get great consistency with most staining methods. I've seen that there's a lot of variability in how things will stain depending on how they're treated. So if I go and get a museum specimen, there's no guarantee that the stain is going to perform the same on specimens within, different series. If one of them was collected in ethanol, another one was collected with ethyl acetate treatment or cyanide, versus something that was stored in Bowen's fluid first. So that makes it a bit difficult. And for most things, the only place to get specimens, I mean, if you're going to do really systematic surveys, the only place to get specimens is the museum collections, unless you go out and collect it yourself. Which poses something of a problem, unless you stick just with commercially available things, which I guess is another matter altogether.
So you also mentioned that there was a distinct difference in the structure of the cuticle, very basal arthropods versus more advanced, which is very exciting because it shows the potential of being able to trace the evolution of more complex structures.
That's a very interesting point. I've looked at fossils of things like trilobites and Loboptera to see if their cuticle has any of these features in it, like the helicoidally arranged stuff. And in trilobites, you actually see it. It's there. They have the same kind of cuticle. So this obviously happened pretty early on. I think it's part of what's allowed insects to be so successful compared to like the Onychophora or some of the tardigrades. Tardigrades are pretty successful already, but they're so small, their cuticle isn't as complex as with insects. So it's a whole different thing. The Loboptera, I didn't see any of that in there, but I mean, not that many specimens with cuticle available. So it seems to have been something that happened really early on though. So that's an interesting line of research that hasn't been touched. Let's see, if I can write down in chat. Non-technical question. Can you tell me if anyone you worked with at Purdue has this sophisticated electron microscopy equipment north of Indianapolis and work with the Purdue Entomological collection? I might be interested in learning more about it. The guy that I work with, his name is Nicholas Chawla. He's in material science. They're still out there. And yes, they have an electron microscope that they use for looking at nano indentations in there, in the things that they perform mechanical testing on. Oh yeah, I gotta go. What else? Those were already there. Is there anybody else in the chat who has a question that I haven't gotten to already? Oh, spell his last name. Sure. There we go.
That was set just as downloader.
Oh yeah. Whoops. Oh. Yeah. That's the one. Anybody else? Let's go twice. All right. Thanks so much for your.
Who's our speaker next?
Jonah Ulmer.
Jonah Ulmer.
I don't know what his topic is.
I bet it's going to be something awesome.
Something kind of not.
It's
Something
it's
kind of
also
awkward.
going to be about insect morphology, except we're going to go to the other side of the epidermis. We're going to talk about endocrine gland and the problem with homology and that.
Excellent. Excellent.
Cool. Might mention, Gary, there's 21 Zoom participants.
21 Zoom participants, and we had how many in here? 18?
Be on the sheet.
It'll be on the sheet. Did everyone here present sign the sheet? If not, please sign it. It's on the pizza box up here. Okay, can I get a motion to close this meeting of the Entomological Society of Washington? Second? It's carried. Thank you, everyone. See you next time.